Introduction. Welcome, medical students, to today's histology lecture on the topic of compact bone. In this comprehensive script, we will delve into the intricate details of compact bone, exploring its structure, function, and the vital cellular components that contribute to its overall integrity. With a focus on the various aspects of compact bone histology, we aim to provide you with a thorough understanding of this fascinating tissue. So, let us begin. Compact bone is composed of haversian canals, which house blood vessels, lymphatic vessels, and nerves to nourish bone cells. Canaliculi connect these cells in the central canal for nutrient exchange. Lamella are layered parallel to the bone's surface, providing strength. Interstitial lamella fill gaps, maintaining structural integrity. Compact bone supports and protects the body. In the haversian systems of compact bone, osteoblasts produce and deposit bone matrix proteins, such as collagen and osteocalcin, which strengthen the bone through mineralization. Osteocytes, located within lacunae, form canaliculi to communicate and exchange nutrients and molecules with neighboring cells. Now, let us dive deeper into the structural and functional characteristics of osteons, the primary building blocks of compact bone. Each osteon consists of concentric lamella surrounding a central haversian canal. The interstitial lamella are positioned between complete osteons, reinforcing the bone structure. Running perpendicular to the long axis of the bone, you will find an intricate network of perforating canals, also known as Folkman's canals. These channels connect adjacent haversian canals, allowing for blood vessels, nerves, and other cellular elements to penetrate the osteon, facilitating communication and nutrient exchange. Perforating canals, as mentioned earlier, provide avenues for blood vessels, nerves, and lymphatics to penetrate the compact bone. These canals are lined with endosteum, a specialized connective tissue layer that covers the inner surfaces of bones. The presence of perforating canals enables efficient nutrient delivery, waste removal, and regulation of bone tissue metabolism. The arrangement of perforating canals is not random but follows a consistent pattern. They usually run perpendicular to the surface of the bone, connecting the external surface to the haversian canals within osteons. This arrangement ensures adequate blood supply, preventing local ischemia and promoting healthy bone physiology. Osteocytes, as mentioned earlier, play a vital role in the communication and cellular functions within compact bone. Through their extensive network of canaliculi, they establish direct connections with neighboring osteocytes. These intricate cellular processes are responsible for various functions, including cell-to-cell communication, exchange of vital substances such as calcium ions, and mechanotransduction. Mechanotransduction refers to the process by which mechanical forces stimulate biochemical signaling within bone cells, regulating bone remodeling and adaptation. In addition to the osteons and osteocytes, the endosteum and periosteum significantly contribute to compact bone's maintenance and repair. The periosteum, the outer connective tissue layer covering the bones, facilitates bone growth, repair, and serves as an attachment site for tendons and ligaments. On the inner surfaces of bones, we find the endosteum, which lines the medullary cavity. Endosteum contains specialized cells, including osteoblasts and osteoclasts, that actively participate in bone remodeling and mineral homeostasis. The presence of an extensive network of blood vessels is crucial for the survival and optimal functioning of compact bone. These vascular structures, found within the haversian and perforating canals, ensure the delivery of oxygen and essential nutrients to bone cells and the removal of metabolic waste products. The intricate vascular supply of compact bone aids in maintaining the tissue's physiological functions, supports its repair processes, and allows for rapid response to injury. Compact bone formation in flat bones, such as the skull, occurs through a process called intramembranous ossification. In this process, mesenchymal cells differentiate into osteoblasts, which begin to secrete the extracellular matrix. As the matrix calcifies, the osteoblasts become surrounded and eventually entrapped within lacunae, differentiating into mature osteocytes. The successive layers of compact bone are formed through the deposition of lamella, resulting in the characteristic organization seen under the microscope. Bone remodeling, a dynamic process, occurs throughout our lives and involves the coordinated action of osteoblasts and osteoclasts. Osteoblasts contribute to bone formation, synthesizing and depositing bone matrix proteins. In contrast, osteoclasts are responsible for bone resorption, releasing enzymes, such as acid phosphatase and collagenases, that break down the mineralized matrix. This continual balance of bone formation and resorption ensures the maintenance of bone strength, adaptation to mechanical demands, and the repair of micro damages that occur during daily activities. Haversian canals, as mentioned earlier, are central passageways within osteons that contain blood vessels, nerves, and loose connective tissue. Around these canals, Concentric layers of lamella are deposited, forming the characteristic structural unit of compact bone. The presence of haversian canals allows for efficient transportation of nutrients and oxygen, facilitating metabolic activities and maintaining the overall health of the bone tissue. Bone modeling and remodeling processes are tightly regulated by various factors to ensure the balanced turnover of bone tissue. Mechanical stress, hormonal factors, and growth factors influence the activation of osteoblasts and osteoclasts, orchestrating the dynamic remodeling process. This continuous remodeling allows bones to adapt in response to changes in mechanical loading, repair micro damages, adjust mineral homeostasis, and support bone growth during development. Finally, let us explore the biomechanical properties of compact bone that make it an incredibly resilient and durable tissue. Compact bone exhibits anisotropic behavior and has varying degrees of mineralization and organization depending on its location within the skeletal system. The arrangement of collagen fibers and hydroxyapatite crystals within the lamella contributes to the compressive and tensile strength of compact bone, allowing it to withstand external forces and maintain its structural integrity. Understanding compact bone histology is important for comprehending the strength and stability of our skeleton. The intricate structure and cellular components of compact bone are crucial for bone health and diagnosing disorders. Thank you for watching.